Welcome to Plant Reactome module of Gramene database. I would like to tell you a little bit about our data model. We are able to host classical enzymatic and transport reactions in our pathway database where we have input molecule and output molecule, and the reaction is either catalyzed by an enzyme or by a transporter protein. In addition to that, we are able to show various other type of reactions such as transitions, such as associations, dimerization, dissociation, and so on. The researchers can access the pathway database from Gramene homepage directly, or they can come to plantreactome.org.gramene. Here they can find various information associated with this database, a tutorial. If they click on Browse Pathways, it will open a browsing window. This left-hand side panel has pathways organized based on ontology concept and enzyme functions. Up here, users can select a given species. Here is Oryza sativa here, for example. This particular window allows, these particular icons allow a differential display of this particular window. This top hand side panel shows a pathway using eukaryotic cell model. Here, the example Jasmonic acid signaling pathway is occurring in two subcellular compartments. Partly seen here is cytosol, and then here is a nucleus. On the bottom panel, we show associated information, a summary overview, and so on. And here, various links to external database such as UniProt, Ensemble, and so on are also be accessed. This is again example of expression data that is available for rice. This is tissue-specific expression data for rice that is fetched from EBI-ATLAS project. And here users can see expression of various enzymes associated with a given pathway. In addition to reference species rice, we also host pathways, projected pathways for 58 plant species, including maize here. We update our database quite regularly, so you can visit it by looking into content and release summary note for updated pathways and updated species. This is an example of maize. Here again, Jasmonic acid signaling pathway is shown here. It is projected for rice, happening in two compartments. On the bottom panel, you can access information associated with this particular pathway. If you look into the molecules and various proteins, you can click on a protein and then access a MageGDB page where you can have a gene model information, access to genome browser, and expression data available for a given gene. Users can compare pathways between reference species rice and their favorite species such as maize. This is what you get. So this is Jasmonic acid signaling pathway, a familiar pathway, and here we have superimposed maize pathway, projected pathway over the rice pathway. If you have blue box, it means that we are not able to map currently any maize gene to this particular enzyme. If you have a partial yellow box or full yellow box, which means that we are able to identify maize homologs for that are mapping to that particular enzyme. This is a good opportunity for maize researchers that they can use rice gene as a query and look for a corresponding maize gene. Another example of IAA biosynthesis pathway superimposed on rice pathway. Users can also upload their data set and analyze omic data. I use tissue-specific expression data here from Shekhon et al., a publicly available data. If you analyze, then you have a choice. You can view pathways using a broader category such as carbohydrate metabolism and see how many pathways under this particular category are differentially regulated across your samples. Here, for example, this is a sample leaf sample. If you look into this orange box, and you can use these arrows to go backward and forward across your various treatments or samples. You can look into an individual pathway such as Starch biosynthesis pathway here. And here, at least 3 enzymes of this pathway are differentially expressed across tissues. If you click on a particular enzyme, you can see genes associated or mapped to that particular enzyme and their differential expression, which is especially important in identifying candidate genes, considering that plants have significant gene duplication, multiple uh, genes that encode for the same function. Again, another example of Kelvin cycle genes and their differential expression. Also, Jasmonic acid signaling pathway differential expression across various tissues, such as embryo here. Users can also upload metabolomic data and pathway in context of pathways. If you have any questions, you can contact us. Finally, we would like to acknowledge funding from NSF and various members of Gramene team and our collaborators from Cold Spring Harbor, USDA, EBI, Oregon State University, Reactome, and SBB. Thank you very much.